Hi and welcome. This is the presentation of Group 1 of the mini project. The group is composed of Dimitra, George, and me, Rafael Silva. So what we prepared for today is that we'll go through the problem that we have, the data sets, the criteria for grading, the implementations, and I mean, we have 2 implementations to meet criteria 5, and then some final words. The project that we selected to work was a image classification problem for detecting brain tumor in images with the help of deep learning and ANNs. The original data sets that we used are those two. So they are from Kaggle. And yeah, I mean, you can access them if you'd like to. And then we have also the repo for the project. I can just open that. This is the project. We have implementation 1 where we find the notebooks that we implemented, but also some results, and then the data sets that we used. And then we have implementation 2 with basically the same structure. We have the notebooks. Here are some notebooks for pre-processing, some of the runs, and then the data sets. Back to the presentation. The criteria for grading was very specific. We tried to tackle all the points, and as we present the notebooks, we will address the criteria there. So I'll start with the implementation 1, and then I'll hand over to Dimitra and George, and then they will walk you through the implementation 2. Yeah, so let's go there and open notebook. I can explain a little bit how we structure this. We have one template where we have a CNN with 3 layers and one with 4 layers. The idea of using this file as a template, we just call that, is because we wanted to speed up run, like runs. So we just made copies. It's not the best way of working, but due to limited resources, we made some copies, and then we split the runs so that we could have more experiments run. But yeah, I mean, here is the notebook itself for 3 layers. Let's see here, 3 layers. And then we install some libraries. We have some, we read the files. You see how many files we have in each of the folders, like the training set, the test set. You have some hyperparameters for the training. And here we are applying pre-processing, which we use the data augmentation. So here are some of the images. There. Here we have the architecture of the model. And then again, in there we added 3 layers. Also, as a criteria point, we use cross-validation. And we have also 2 seeds for this. In this case, we had 3 seeds there. And we are, we are like collecting metrics, accuracy, precision, recall, area under the curve, and F1 score. I mean, these results here, they are a bit strange, but that's because it's the template. We were still working on that when we run this specific notebook. But as the runs, the real runs, they are more significant. Yeah, then we have one TensorBoard there also that we could explore a little bit of the outputs there. So yeah, if I go back here to the folder, then we have the runs. I can open one actual run there just briefly. And yeah, I mean, as you can see here, a bit more down, we have some more real runs of the templates. But yeah, I mean, we have different settings. Like you are using two different data sets also. But one thing that I would like to walk you through is the actual runs. So for data set 1, we set different parameters. For example, like a number of epochs, number of hidden layers, number of seeds, the optimizer, and then we have some performance validation there, performance metrics during the training. For yeah, after the training, we also applied validation on the test, the data. So we have like this table, which is also available on GitHub. Out of our runs, I mean, what we tried to mix was the optimizer, like you see here that we used 3 different. And then we have the number of epochs. See, sometimes we run with 10, sometimes with 15. And just like trying to follow the criteria for the assignment. So again, this file is on GitHub also there, together with our presentation, together with our project. But one thing to mention here is which values we got the best results. So the best results on validation data that we got was 85% of correct answers. And that was with this configuration here. We had 15 epochs, we had 3 layers, we had 2 seeds, and we use other grade there. However, the best F1 score during training was with this other setting here. So that, yeah, I mean, they were not the same, which is expected. I mean, this is training, this is validation. So yeah, this was the best one, F1 score. Yeah, and this was for dataset 1. We have another run for dataset 2. And following the same mixing parameters there, what we could achieve as best validation was 62.5%. And it had 3 layers, other grad, 2 seeds, 10 epochs. And also this one got the exact same result. And the best F1 score that we got was for this configuration here. So now I'll hand over to my colleagues, my team mates, and yeah, they walk you through the other solution that we implemented.
Let's now discuss something more specific about the preprocessing we applied for our datasets. We used the two-step procedure for the reprocessing process. First, for each of the datasets, we checked for duplicate images. This was due to the datasets including a lot of duplicates. Then we autocropped each image based on the bounding box, because the datasets contained images that had different dimensions and ratios, and the image was placed in other parts of the image. Also, we have included a selection, a random selection algorithm to select a random subset of the original dataset. This was due to some datasets having a lot of images, and we wanted to select only part of these images for our training purposes. Also, in the main training notebook, we created an image data generator that could normalize the image, and we rescaled the images to the required input size for our models. We also developed another notebook that was used only for datasets 5 and 6, but this was much simpler, as these datasets were already split into train and test directories, and it was not needed to move files to other directories, and we weren't supposed to make a new folder structure. There were not as many duplicates. However, there were some, so these were deleted, and we didn't need to autocrop them. About our custom convolutional neural network, we wrote a function that creates a custom pyramid-shaped convolutional neural network for different number of layers, 3, 4, and 5, different kernel size, 3 by 3, 5 by 5, and 77. And we used different optimizers like Adam, SGD, and AdaGrad. We used 30 epochs, and we implemented early stopping. For our first run, we trained and evaluated the model for different combinations of following hyperparameters, 108 combinations in total, and data sets. We used different seeds, 42, 143, and 82. We used different optimizers, different number of layers, different sizes of kernels, and 2 datasets. We set the parameters, training history, and evaluated the test metrics, loss accuracy, precision, recall, F1 score, and the training time of each combination in that frame, and presented the results in the notebook. It seems that the Adam optimizer seems to perform the best. The number of layers and kernel size impact is not as significant, and we came to the conclusion that the performance on the second dataset seems to be worse than on the first one. This is probably due to dataset 2 containing 2 different types of images, both top and size brain scans, which would require significantly larger dataset compared to dataset 1, which contain only top views. For the second run, we used also k-folds, and we modified the code to implement the stratified k-fold specifically. We rerun the code for the following combinations, different seeds, the same seeds as before, the Adam optimizer, we used 3 and 4 layers, 3 by 3 and 5 by 5 size of kernels, and different number of folds, 4 and 6. We once again saved the parameters in the training history, and we evaluated the test metrics in training time. It seems that the Adam optimizer performs the best. The number of layers and kernel size impact is not that significant, and it seems that the performance is worse in dataset 2. This is again due to dataset 2 containing 2 different types of images, both top and side spring scans, which would require significantly larger dataset compared to dataset 1, which contain only top views. The performance compared to the simple no k-fold is not dramatically improved. For our third run, we attributed the apparent low contribution of k-fold to the small size of datasets. For this, we ran again the k-fold code, this time on the full preprocessed dataset 2. We use all of the images instead of 6% of the images we selected for the second dataset, with 2 different seeds, 42 and 133. We used Adam, 4 layers for the CNN. Our kernel was 3 by 3, and we used 5 volts. This took a long time to execute. It took about 2.5 hours for each seed, and the average test accuracy for the 2 seeds was calculated at about 95% compared to the previous 63%, which indicates that the model needs a larger dataset to be trained effectively.
We will now demonstrate the use of transfer learning on our 2 datasets. For this task, we will use 2 different pre-trained models, one called ResNet 50 and the other is called VGG19. First, we try ResNet 50 on our 2 datasets. That is dataset number 1, containing approximately 250 images, and a subset of dataset number 2, containing 6% of the original approximately 4,000 unique images, and that makes it 243 images. So the size is comparable between the 2 datasets. We use 3 different seeds, and in order to do that, with our names, we created a custom function that creates numerical seeds based on string datasets, because NP random seed doesn't support string seeds. We train the 6 combination and optionally save the trained models to use later using Pickle. We can plot the training histories from the Pickle files. And on this slide, you can see an example of such a plot using the training for the seed Georgios and dataset number index of dataset number 1, that is the dataset number 2 of 6%. We can then evaluate the trained model using the test subset, and we can plot the confusion matrix as well as the performance report. In which case, for example, for the training for seed Georgios and the index number 1, we see that the average, the test accuracy is approximately 88%, and the performance metric, different performance metrics such as precision, recall, and F1 score are tabulated in the report. We can also tabulate on the same slide the test accuracies we got for the 6 different combinations of seed and indexes and datasets, and the average of these 6 runs is approximately 87%. Because we suspect that a big factor for the relatively low performance is the number of images we used, we ran the whole, the same code, this time for the whole dataset number 2, which includes all approximately 4,000 images. we chose the seed that performed the worst in our previous run, which is Dimitra, with approximately 84%. The training took approximately 1.5 hours, and we see that the test accuracy on the whole dataset is now calculated at approximately 97%, which is significantly higher. This indicates that the number of images is a crucial parameter in our problem, and having a low number of images in dataset number 1 is a limiting factor. In order to evaluate how big this effect is, we choose 2 random datasets from Kaggle containing approximately the same number of images, approximately 200 to 300 images, and calculate the performance for using the same code. This time using the course's code as the seed. We see that the performance that we get is between 92 and 98%, almost 99, which indicates that learning to differentiate the brain tumors, it's a much more difficult task than differentiating between cats and dogs, or even COVID X-rays. This suggests that in order to get a good result, we need a relatively high number of images. We then demonstrate the use of VGG19 pre-trained model on the same 6 combination of datasets and seeds, and we get the same graphs and reports. And if we tabulate the 6 test accuracies, this averages to approximately 73%. This means that the ResNet 50 based model outperforms VGG19 in all average test accuracies criteria, on average test accuracy, minimum and maximum. This leads us to suggest that the ResNet 50 based model is the model of choice to go, because it achieves better accuracy compared to the VGG based one. And although it has comparably, even slightly higher accuracy than our custom model, it runs much faster, approximately 2 to 3 times faster than the custom model we tried. Our custom model has shown that it can reach comparable accuracy to VGG19. However, still, VGG19 has a much shorter training time, which makes VGG19 the second better model, and our custom model the worst performing one. We can then show how we met the grading criteria. We have used pre-processing of duplicates, image cropping, and normalization. We demonstrated exploring different hyperparameters such as number of layers, kernel size, and the optimizers, and evaluated the impact of them on model performance. We used cross-validation and evaluated the impact on the performance increase. We used different seeds, numerical and string, and monitored, recorded, and plotted performance metrics during training. And we also used the code on multiple datasets and subsets of them, and compared the performance between them. And we have also compared the performance of different models and concluded the recommended model to go with. And this is the end.